What picture comes to your mind when you hear the word algae? Yes, they are the green structures floating on ponds. Most algae are green in color because they possess chlorophyll. Hence, they are autotrophic, as they can photosynthesize and prepare their own food. Some algae are even brown or reddish color, but that is because they possess other pigments in addition to chlorophyll. Algae, by means of their photosynthesis, increase the dissolved oxygen level in their surroundings. They are primary producers in most aquatic ecosystems. Small fishes and similar creatures feed on algae. Algae are simple, thalloid, and mostly aquatic, found both in fresh and marine water. They are found in moist land regions too. Some algae form symbiotic associations with fungi, as seen in lichen. There are microscopic unicellular algae such as the Chlamydomonas and giant forms such as the kelps. Some, such as volvox, form colonies. They may be filamentous, for example, Eulothrix and Spirogyra. Algae show vegetative, asexual and sexual modes of reproduction. Asexual reproduction is by means of spores. They are generally motile flagellated spores. In sexual reproduction, the gametes may be flagellated or non-flagellated. They may be similar or dissimilar in size. Therefore, sexual reproduction may be oogamous, where one large non-motile female gamete fuses with a smaller motile male gamete. It may be isogamous, where the two fusing gametes are similar in size. It may be anisogamous, where the fusing gametes are dissimilar in size. The life cycle of algae show alternation of generation, where the gametophytic phase is the dominant phase. Algin, carragin, agar, and single-cell proteins are products of commercial importance obtained from algae. Chlorophytes, belonging to the class Chlorophyceae, are green algae. The pigments chlorophyll A and B are dominant in them. They possess chloroplasts which vary in shape, being either disc-shaped, cup-shaped, plate-like, reticulate, or ribbon-shaped. Chlorophytes also possess storage bodies called pyrenoids that are present inside the chloroplasts. Pyrenoids store protein and starch. Chlorophytes have strong cell walls made of an outer layer of cellulose and an inner layer of pectose. Most chlorophytes live in fresh water, but some are found in marine habitats also. Chlorophytes may be unicellular, such as the Chlamydomonas, or multicellular, such as Chara, colonial forms such as volvox, or filamentous, such as Spirogyra. On glaciers at high altitude, you might see something called watermelon snow. This is because of a green alga, Chlamydomonas nivalis, that forms dense algal blooms in such snowfields. It produces reddish pigments, which gives red color to the snow. Most chlorophytes reproduce sexually by isogamy, oogamy, or anisogamy. Spirogyra can reproduce sexually by conjugation. Chlorophytes reproduce vegetatively by fragmentation. They also reproduce asexually by means of flagellated zoospores. Chlamydomonas, volvox, Eulothrix, Spirogyra, and Chara are some common examples of green algae. Phaeophytes, belonging to the class Phaeophyceae, are brown algae. They are the largest and most complex algae. Phaeophytes are multicellular and mostly marine. They comprise most species that are commonly called seaweeds. Some are simple branched filamentous forms such as ectocarpus, while some are profusely branched, for example the kelps, which can be as long as 100 meters. The phaeophytes are brown because of the carotenoids present in their plastids. Their color ranges from olive green to various shades of brown. The color depends upon the amount of fucoxanthin, a xanthophyll pigment, present in them. Brown algae store food in the form of complex carbohydrates such as laminarin or mannitol. Their vegetative cell walls are made of cellulose with an outer gelatinous covering of algin. They are mostly attached to a substratum by a small organ called a holdfast. They have a stalk which is called the stipe. Their photosynthetic organs are leaf-like structures called fronds. Brown algae reproduce vegetatively by fragmentation and asexually by the formation of zoospores. Zoospores are biflagellate, pyriform or pear-shaped, and have two unequal laterally attached flagella. Phaeophytes are also capable of reproducing sexually by isogamy, anisogamy, or oogamy. The gametes are pyriform with two laterally attached flagella. Examples include ectocarpus, dictyota, laminaria, sargassum, fucus. Rhodophytes belonging to the class Rhodophyceae are red algae. They are reddish in color because of a pigment called R-phycoerythrin, which masks the green color of chlorophyll. Most red algae are found in marine habitats. They are present in surface as well as deep waters. They are multicellular and some of them have a complex body organization. The storage polysaccharide in red algae is Floridian starch, a polysaccharide similar in structure to amylopectin and glycogen. Vegetative reproduction in rhodophytes takes place by fragmentation. They reproduce asexually by means of non-motile spores and sexually by non-motile gametes. Sexual reproduction occurs by oogamy. Common examples of red algae are Polysiphonia, Porphyra, Graciliaria, and Gelidium. Walking along a road during rainy weather, you might have noticed a greenish growth on walls. You call it moss. Moss comes under the division Bryophyta of kingdom Plantae. Bryophytes include mosses and liverworts, which grow on moist, shady surfaces. They need water for reproduction and hence are called the amphibians of the plant kingdom. The male gametes require a film of water to swim and reach the female gamete. The plant's body is a haploid gametophyte. It may be simple thalloid in appearance, or it may be differentiated into stem-like, leaf-like structures and rhizoids. However, remember that these are not true leaves, stems, or roots, as they lack vascular tissue. The rhizoid helps the plant's body to remain attached to a substratum and absorb water and minerals. Bryophytes show alternation of generations. The life cycle of a bryophyte is complete when the plant has passed through both stages, the gametophyte and the sporophyte. The gametophyte reproduces sexually by gamete formation. Male gametes are produced in the male sex organ, the antheridium. Male gametes are biflagellate and are called antherozoids. Female gametes are produced in the female sex organ, the archegonium. A single egg is produced by the archegonium. The fusion of the male and female gametes results in a diploid zygote. The zygote gives rise to the diploid sporophyte. The sporophyte body is differentiated into foot, seta, and capsule. The capsule contains haploid spores. The spore germinates to give rise to a haploid gametophyte. And the life cycle continues. The life cycle of bryophytes shows both haploid and diploid stages, and hence they are said to show alternation of generations. Liverworts are generally found along the banks of streams, in damp soil, and on tree barks in dense forests. Riccia and Marchantia are examples of liverworts. Liverworts have a flat green, lobed or forked thallus. The thallus is dorsiventral. This means that it has an upper side and an underside. The thallus is attached to the substratum by thin rhizoids. Liverworts reproduce asexually by fragmentation of thalli or by specialized structures called gemmae. Gemmae are asexual buds which develop in structures called gemmae cups present on the thalli. The gemmae detach from the parent body and develop into new individuals. For sexual reproduction, male and female sex organs are produced by the gametophyte either on the same thalli or on different thalli. An upright structure called the archegoniophore bears the archegonia, and an antheridiophore bears the antheridia. The antheridia and archegonia bear the male and female gametes respectively. The sporophyte formed after fusion of male and female gamete has a foot, seta, and capsule. Spores are formed in the capsule by meiosis. The spores germinate and give rise to new individual gametophytes. Mosses, unlike liverworts, have an upright stem-like structure. Funaria, Polytrichum, Sphagnum are a few examples of mosses. Leaf-like structures are spirally arranged around the stem. Rhizoids in the basal region attach the moss plant to the substratum. The first stage in the life cycle of a moss is the protonema stage. The protonema develops from a haploid spore. The second stage is a leafy stage, which forms from a lateral bud on the protonema. It consists of leaves arranged spirally around an upright axis. It is attached to the soil by branched rhizoids. This stage also bears sex organs. The sex organs are produced at the apex of the leafy shoots. After the fusion of male and female gametes, a sporophyte is formed. The sporophyte has a foot, seta, and capsule. Spores are formed in the capsule by meiosis. The spores germinate and give rise to new individual gametophytes. Gymnosperms are a group of seed-bearing, vascular and non-flowering plants. The word gymnosperm arises from the ancient Greek word gymnospermos, meaning naked seed. The unfertilized ovule and also the seed, which develops after fertilization, both remain exposed and are not covered by the ovary walls, as are the seeds of flowering plants. Gymnosperms are either tall or medium-sized trees or shrubs. The sequoia or redwood tree is a gymnosperm and is the tallest tree in the world. They are broadly classified into conifers, cycads, knee-tails and jingo. The jingo is considered to be a living fossil and originated approximately during the Permian, which means that it dates back 270 million years. Gymnosperms have taproots which exhibit different types of symbiotic associations. Pinus roots are symbiotically associated with a fungus known as mycorrhiza, whereas cycas bears coralloid roots associated with a nitrogen-fixing strain of cyanobacteria. Stems are branched in Pinus and unbranched in cycas. Leaves may be simple and resemble scales, needles, or broad blades. Or they may be compound with thick, leathery leaflets borne in a feather-like, pinnate arrangement on a main axis. Gymnosperm leaves are modified to reduce their surface area, while the thick cuticle and sunken stomata prevent water loss. The main plant body of a gymnosperm is a sporophyte. They are heterosporous, producing two spore types, microspores and megaspores. The microspores and megaspores are borne respectively on male and female cones or strobili. In Pinus, the male and female cones are present on the same tree, while in cycas they are borne on separate trees. The male cones are composed of spirally arranged rows of microsporophylls containing microsporangia, within which the microspores are produced. These microspores form the pollen grains which represent the reduced gametophyte. Similarly, the female strobilus or the ovulate cone bears the megasporophyll, which contains the ovule or megasporangium. The ovule is a composite structure covered by an integument. In the ovule, one of the cells of the megasporangium or nucellus differentiates to form the megaspore mother cell. The megaspore mother cell undergoes meiosis to form 4 megaspores, one of which eventually develops into a multicellular female gametophyte. The female gametophyte contains two or more archegonia or female sex organs, all retained within the megasporangium or nucellus. The pollen grain is released from the microsporangium and comes into contact with the ovule borne on megasporophylls. The pollen tube carrying the male gametes grows towards the archegonia in the ovules and discharges its contents near the mouth of the archegonia. Following fertilization, the zygote develops into an embryo, which eventually grows into a new plant. Angiosperms are a large group of plants with great diversity of appearance. The smallest flowering plant, Wolffia, is almost microscopic, while the tallest, Eucalyptus, measures up to 100 meters. Angiosperms are vascular and flowering plants. The main plant body of angiosperms is the sporophyte. Their ovules and pollen grains are developed within specialized structures called flowers. In angiosperms, the seeds are enclosed by fruits developed from the ovary. Angiosperms are classified as monocotyledons and dicotyledons on the basis of the number of cotyledons present in the seeds. Monocotyledons or monocots have only one cotyledon in each seed, while the dicot seeds are characterized by two cotyledons around each seed. When a monocot seed germinates, it produces a single leaf, but when a dicot germinates, it produces two seed leaves. Monocots usually have fibrous roots, while dicots have taproots. In monocot roots, radial vascular bundles generally exhibit polyarch condition, whereas dicots generally have tetrarch condition. In a monocot stem, the vascular bundle is scattered throughout the ground tissue, while in a dicot they are arranged in a ring. Monocot leaves have parallel venation, while the dicots have reticulate venation. In monocots, the floral parts are seen in multiples of 3, while in dicots they are present in multiples of 5. In monocots, the pollen grains have a single pore, while in dicots they have 3. In monocots, there is no secondary growth, while in dicots, secondary growth is often seen as growth rings. In angiosperms, the flowers contain the male and female sex organs. Both the sex organs are present either in one flower or in different flowers of the same plant. The male sex organ of a flower is the stamen. Each stamen consists of a thin, slender filament with an anther on its top. Anthers undergo meiosis to form pollen grains. The female sex organ of a flower is the pistil or the carpel, though a pistil may also be made up of many fused carpels. The pistil is composed of an ovary, a style, and a stigma. The ovary is composed of one or many ovules. In the ovules, the megaspore mother cell undergoes meiosis to form the haploid female gametophyte called the embryo sac. The embryo sac contains a 3-cell egg apparatus composed of an egg and 2 synergids. The embryo sac also contains 3 antipodal cells and 2 haploid polar nuclei, which eventually fuse to form a diploid secondary nucleus. Pollen grains are dispersed from the anthers and carried to the stigma by a process called pollination. Various agents such as wind, water, and animals mediate pollination. Following pollination, the pollen grains germinate to form the pollen tube, which then reaches the ovule. The pollen tube enters the embryo sac and releases the two male gametes, one of which fuses with the egg cell to form a zygote, while the other one fuses with the diploid secondary nucleus to form the triploid primary endosperm nucleus, or PEN. This unique process of fertilization in angiosperms is called double fertilization. The synergids and antipodals degenerate after fertilization. The zygote develops into an embryo and the PEN develops into endosperm, which provides nourishment to the developing embryo. In the course of the fertilization and development of the embryo, the ovules develop into seeds and the ovaries develop into a fruit. Plants exhibit two multicellular adult forms during their life cycle, one diploid and one haploid. They give rise to and alternate with each other in their life cycle. This is known as alternation of generation. The diploid plant body is known as a sporophyte with two N chromosomes, while its haploid counterpart is known as a gametophyte with N of chromosomes. In plants, both the haploid and diploid cells can divide by mitosis. The haploid plant body, the gametophyte, produces haploid gametes by mitosis. Following fertilization, these haploid gametes form a diploid zygote, which eventually grows into the diploid sporophytes. The mature sporophyte produces haploid spores by meiosis. These spores, in turn, divide by mitosis to form a haploid gametophyte plant body once again. This pattern of alternation of generation is seen in all groups of plants. On the basis of the dominant phase of their life cycle, plants are of three types: haplontic, diplontic, and haplodiplotic. In most of the algae, such as Chlamydomonas, the main photosynthetic plant body is a free-living gametophyte. The unicellular gametophyte divides by mitosis to produce haploid gametes of plus and minus strains. Two haploid gametes of opposite strains fuse to form a diploid zygote, which eventually undergoes meiosis to produce haploid spores. There is no free-living sporophyte stage. The spores grow into the haploid gametophyte, or the main plant body of the Chlamydomonas. This pattern in which the dominant plant is haploid is known as the haplontic life cycle. Gymnosperms and angiosperms exhibit a type of life cycle wherein the dominant photosynthetic plant body is the sporophyte. This pattern, where the dominant plant is diploid, is known as the diplontic life cycle. The diploid sporophyte produces haploid gametes through gametogenesis. There is no free-living gametophyte phase, which is represented only by a few cells that remain inside the body of sporophytes. Following fertilization, the haploid gametes form the diploid zygote, which eventually grows into the dominant plant body or sporophyte. Bryophytes and pteridophytes exhibit an intermediate type of life cycle called the haplodiplontic life cycle. Here, both the haploid and diploid phases are visibly dominant in the plant body. In pteridophytes such as ferns, the well-developed photosynthetic phase is the sporophyte, while the gametophyte is less developed, forming a small flattened thallus. The sporophyte produces haploid spores by meiosis, which germinate into the haploid gametophyte body. The gametophyte in turn produces haploid gametes by mitosis, which form the zygote and, following fertilization, eventually develop into the erect sporophyte. In contrast, in bryophytes such as Funaria, the gametophyte is more developed than the sporophyte and is represented by an independent photosynthetic erect phase. The sporophyte is less developed and is entirely dependent on the gametophyte for its nutrition and anchorage. The diploid sporophyte produces haploid spores by meiosis, which germinate into the main plant body or gametophyte. Gametophytes each produce a sperm and ovum, which fertilize to form a diploid zygote. Following fertilization, the zygote divides mitotically to grow into a diploid sporophyte.